case of Google Earth. So what we tried to do in the Whole Brain Project is try to look at the best examples that are out there for organizing other information and apply that to the brain. So what we've done is we've taken a hint from Wikipedia and created an online wiki for neuroscience called the NeuroLex. This is in collaboration with the Neuroscience Information Framework. And the basic idea is it's built on the same software that runs Wikipedia. It has some extra functionality that allows you to create the kinds of ontologies that I was just talking about, and allows anyone to edit or create new concepts. And to date, it has about 16,000 concepts. And what's really cool about this project is that with zero advertising, we get about 80% of our hits that come in from search engines. So what this means is this is filling in a gap that exists where people are looking for information that they would probably want to find inside journal articles, but in fact they're hitting NeuroLex first. So this is an example of the set of search queries that come up with NeuroLex. So if you search for dorsal root ganglion right now, a NeuroLex link will come up. If you're searching for medial meniscus, a NeuroLex link will come up. And this is just sort of the first 15 terms that come up when the most popular searches that will send you to NeuroLex today. And I think that's pretty exciting because now we're getting about 250 hits a day that are coming in from around the world for people looking for information. And I think what this means is that we're filling in a gap where people are searching Google first rather than search. So who makes sure this sort of data is reliable? Anytime you talk about wikis, people are always wondering, well, it's going to be the Wild West. How do you know that that's actually valuable information? Well, there's several projects that have taken the charge up for doing this. So the Neuroscience Information Framework is one, I've already mentioned that. Also something called the INCF, the International Neuroinformatics Coordinating Facility, is a really important organization that's trying to create standards across the globe for people doing these kinds of information technology management inside neuroscience. And they've also collected together task force of neuroscientists, some of whom you might be familiar with, who are then pointed at things like NeuroLex to curate it and make sure that the data there is actually valuable. But really, the ideal thing is that the people who are making sure this data is reliable is you, the community of neuroscientists who are actually involved in investigating the different systems that you're involved with. It's really, we really need to make sure that these tools get to a level of simplicity so that anybody can take advantage of them, such as you. So I really encourage you to check that out. I talked to you a little bit now, switching over to the where side, about Google Earth. And of course, Google Earth is a system where images that are taken from satellites and cars that drive around, and I think even bicycles, Google bicycles, are all collected together in a single virtual 3D globe, and that allows anyone to investigate a whole range of information that is spatially located, and sometimes even results in a Wikipedia hit. So the multi-scale nature of the Earth is something that I think we're all familiar with, and getting a picture of how that works together is something that Google Earth is very effective at doing, so that we can go across geographic scales. So what we tried to do with a project that we call the Whole Brain Catalog is do a similar thing. So start with images that come from microscopes, put together a 3D virtual brain, and then allow anyone to investigate those pieces and parts with some of those links getting you back to the wiki view of that data. And so what we really wanted to do, and we've been fairly successful with the first beta version, is to do that, so that you can go inside the skull of a mouse brain. So we started with mouse. You can see the regions that are inside of it, and then even drill down to the cellular level and beyond in order to see the different levels of complexity that are there. So that's just a start to that. So the Whole Brain Catalog then it's completely open source. It's a multi-scale tool that's built on top of an open source game engine. So we're using technologies that would otherwise be used for shoot 'em ups for the purpose of science. It's an environment that's intended to allow individuals to share data between, as well as a way to get folks in computational neurosciences who typically build models that are disconnected from microscopy data to have a place to mine so that they can build models that are more realistic and that are based on cell fills and other microscopic data. And it's also an environment for putting together databases that are distributing information on neuroinformatics in a single place. So that's a little bit about the interface. I'll just skip that in the interest of time. So I wanted to point out that the Whole Brain Catalog is based on the Allen Institute's three-dimensional reference atlas. So when you're looking at these shapes and pieces and parts, what we're really looking at are triangular meshes that define the delineations of the different cell groups, cell regions that make up the brain regions of the Allen brain. And so that's an important thing, because one of the valuable things that you can do in the Whole Brain Catalog is you can set down a probe in 3D space and look at different pieces and parts and features that exist in there. So one of the things that you can look at, for example, are you can take a cell that's been situated inside that brain and you can get information from the neural net, such as this. Another valuable thing that you can do is you can share data across scales. So this maybe is an example where we've placed data, we've got data from the Allen Institute as well as, as well as a single cell fill that comes from a 1997 paper that was published in the Journal of Neuroscience that was talking about that individual cell, and also electron micrographic reconstructions that come out of the Ghosh lab from the work of Scott Wilkie, where he was examining the dendritic branch and the spines that are associated with the CA3 dendritic branch. So that's, I think, an example of what data sharing might look like in the future, where all the pieces and parts of the brain are all situated together so that you can smoothly go between cell fills such as this, which then can also have animations that are associated with them in terms of models that have been attached to those cells. The subcellular reconstructions that are situated in the part of that reconstruction where it's appropriate, and even go all the way down to models that are pulled from things like the protein data bank, so you can see what are the potassium channels that are known to be associated with them, and even get the structure back out of them, so you can put that together for a model. In addition, I mentioned that it's connected to the Allen Atlas, so another thing that you can do when you put a spatial probe in this space is that you can get gene expression out of this. So by positioning a little orange sphere at this spot, you're able to right-click and bring up that part of the Allen Institute's Gene Expression Atlas that shows you what are the genes and what are the similarity of genes in that particular region. And what we're working on next is to actually bring those gene expression patterns back out so that you can browse them per gene. So this is an example of a single gene being visualized across the Allen Institute's brain regions, and that's something that we're really working on. So in the interest of time, I wanted to skip ahead past some work on connectivity, talk a little bit about simulations that have been integrated into this environment. So, back in 1995, Terry Sinowski and Zach Manin published a paper discussing a model of neocortical pyramidal neurons, and they were gracious enough to publish that in a way that could be accessed. So what we've done is we've demonstrated that we can pull that into the whole brain catalog, such as this, from a site that publishes models, and we've been able to create a system that allows you to directly simulate and visualize the results of those simulations back in the context of the whole brain catalog. This being another way that we can tie in data that already exists to other published data that come out of microscopy work. A further example of a model that's been published is this one from Eric DeSchutter, who did work. Let's see, actually, get that to run. Did work on creating this simulation of the cerebellar cortex. So looking at the granule cells of the cerebellar cortex, and that's another thing that, again, we situate in the context of the cerebellum within the Allen Institute's brain regions and visualize all within this tool that anyone can access. Okay, so what I wanted to do before I finished was to talk about a real example of using this for science. So as you're all aware, I'm sure, in 2004, the Nobel Prize was handed out for work in the olfactory bulb to understand how olfaction works. And folks have continued on this work at the Scripps Research Institute to try to go beyond just the olfactory epithelium to try to understand the organization of the olfactory cortex and how the glomeruli in the olfactory epithelium project to the olfactory cortex that goes beyond that. So thanks to the work of Jeff Bush, who came in a rotation and engineered an ability to do much more precise manipulation of data within the whole brain catalog, we worked with the Baldwin Lab at the Scripps Research Institute to solve a problem that they were having of being able to integrate neuronal structure together in a single reference brain. So what they did is they, using Neurolucida, they constructed a reference brain of a mouse that was 3 weeks old. This was important because the Allen Atlas brain is a full adult, and they needed to use a 3-week-old brain. So they reconstructed the olfactory regions that they were interested in, and then they started placing their data sets into the whole brain catalog because they needed to synthesize across different individuals in a way that they could see the morphologies of the neurons that they were tracing out all connected together. So this is an example of Dia Ghosh in the Baldwin lab, using these handles that allow manipulation inside 3D space. And what she's doing is she's lining up data sets that they took where they used cell fills in the olfactory bulb, both in 2D and 3D, and lining them up in this reference atlas. When you do this, you get something like this, so to look a little bit more in depth. So here we're looking head-on the olfactory bulb of a mouse that's been projected inside the whole brain catalog. And as we zoom out, we're able to take this data set up here with the handles that it's got, and you're able to select that whole data set as a group, both the 2D and the 3D elements together, and we're sort of rotating around to the side. And in here is the morphology, the neuromorphology that we care about. That's the key of all of this work, is to get those guys put all together. And so what Dia is able to do is to slide that into the reference brain in the place where it is appropriate, use the 2D to match up where that needs to go, and then remove the 2D parts to reveal the 3D structure as it's located inside the olfactory bulb, and then start to ask questions about how do those morphologies relate to the reference brain structures that they've created. And what that results in then, just go into this here, what that results in, oh, this is very dark. Okay, here we go. What that results in, once you've done that several times, is you get all of the cells that you've put together across multiple experiments in a single three-dimensional space, and you can compare that to the brain regions that have been segmented out and ask questions, like I said, about those brain regions. Now, I know this looks messy and like a messy tangle, so the important thing now, of course, is that that all of these cells are digital entities which can be computed upon, and that's exactly what we did, is we then extracted these shapes, asked the computer to tell us a little bit spatially about how they're related to each other, did an endpoints analysis, and we were able to see the distribution of how these cells were located near each other. And this work, I'm happy to say, was just accepted to the journal Nature in a paper that's being published by the Baldwin Lab. And so you'll be able to hear more about the full scope of this in terms of all the experimental details in February.